Open it. You have to finish. Then open it stratified, compressed. Then go to second week. Which one? Second week, week 2. Go down. Maybe yeah. You have to go back. Week 2. Yes. Sorry. Yeah, let's open this. And you have to finish all. So start with simple.
I would press record screen.
Writing it down.
Even my laptop, my Mac, I use mouse. So I'm not good at using this. It's good. Mic is working. Yes.
One last thing.
Fine, right?
It's very casual. Just or something. Whatever. Yeah,
Just like
do
this.
it.
Again? I am kind of nervous. Like,
Okay, I mean,
Right.
You want full screen?
Yes.
I
Better.
consider yourself just talking.
Down.
You talk in the mic, just so I can make sure.
Okay. Bismillahi minash shaitani rajim. Bismillahir rahmanir rahim. So, new place, new environment, no eye contact, so no interaction possible. So today it will be a different strategy. I will take all your questions at the end. Okay? So I have to finish my topics. I need your help. They're all easy now because I assume that you have a good foundation. Most of you have read all those lectures during your weekend. So with this, I will start. Any question that comes to your mind, either please write, keep it on hold till the end. Most of the question I'll try to answer during my explanation. Okay? So because I don't know whether I can see the last one or not, I'll try to take all your questions as I normally do. Whatever number of emails I received during weekend, I started receiving from Thursday evening. So I replied almost all. To some of the emails, I have asked them to come sit with me if they need more explanations. It's up to you. Okay. Now, the fifth type of simple epithelium, it is called as pseudo-stratified epithelium. So because there is no mouse, so I'm finding it difficult. Okay. Now see, as you can see in the diagram, it is an epithelium. And the key point is that if you see in the diagram that all these cells are touching the basement membrane despite having different sizes. This is very important point. All the cells are touching the basement membrane. So it means that they all are in one line. So it is a simple epithelium, point number 1. Now then why it appears stratified? Here you have to apply that information that I gave you on the very first day, that when we see these cells under the microscope, plasma membranes are not visible. When plasma membranes are not visible, so it appears as if the nuclei or of different sized cells are present at different levels, and it appears as if they are arranged in layers. But when we draw lines, when we trace the plasma membranes down to the base, then it becomes clear, no, the appearance is because of the different location of the nuclei in different sized cells. That is why it is giving us a wrong impression. This is why it is called as pseudo-stratified epithelium. You can see here, it is a type of simple epithelium because it consists of one layer of cells. The cells are of different sizes, some are small, and some are large. They all are columnar. Okay, they all are touching the basement membrane. So this is the proof for simple. It gives the wrong impression of being arranged in different layers. So this wrong impression is given, once again I am repeating, by the nuclei of the cells which are present at different levels. Okay, now see, because we are discussing histology in general, epithelium in general, so all different types, they are found in different locations. So at this time, at this particular moment, you need to simply know. Do not try to memorize or learn by heart. Just read. All these sites will be discussed in detail. All these places will be shown to you when we will be discussing those organs. Okay, so pseudo-stratified epithelium is of 4 types. There are 4 variants. You can see pseudo-stratified columnar means there is nothing associated with this. Then second is pseudo-stratified columnar with cilia. Then third one is the most important type. It is pseudo-stratified, it is columnar, it is ciliated, it is having goblet cells. Along with the columnar cells, it is found in the respiratory tract. The epithelium, which is found in the respiratory tract, in bronchioles, in trachea, in bronchi, to expel the mucus, the bulgum, out of the respiratory tract, is this third type, where there are cilia, where there are mucus secreting cells, which are called as goblet cells. And then finally, fourth type of the pseudo-stratified epithelium, it is mainly found in the male reproductive tract. It is associated with stereocilia. So I'll show you slide for each of these types without giving details, right? Look at this. This is first of the pseudo-stratified, where it is columnar without any association. You can see here, you have to start from here. You have to start from lumen. Look at this epithelium. At this point, it gives the wrong impression. But see, there is nothing associated with this. But one thing I'm going to show you, you please remember, and then I'll explain. Can you see this part? This part, can somebody come and help me in finding the laser pointer? I'm not good at this. Anyone, please? Anybody, just come forward. That's what I'm trying. Come quickly, anybody. Faisal. Now you have your new BLC. Partially I'm announcing. Faisal is mashallah handsome.
Thank you.
Need to sometime please somebody, because I need a party. They have promised. Faisal, you promised. Party. 2, okay? Now, you see, this is the open area. And look at this. This point is important. Then I will keep explaining it. The upper part of the cell in pseudo-stratified epithelium, the upper part of the cytoplasm has no nuclei. Has no nuclei. All nuclei are trying to be towards the base. They have a tendency to lie towards the base. Nothing is here. This is a very important property. I will name it. Okay. Now, second type of pseudo-stratified epithelium, but this time it is associated with cilia. Look at this epithelium. Here it's confusing and giving us the wrong impression. But once again, look at this property where the upper part of the cytoplasm of all the cells are empty. All the cells are polarized towards the base, leaving behind the upper part of the cytoplasm empty. So here we will use the term polarity, polarization toward the base. Plus, the presence of cilia is the second important point. Cilia. So its name is pseudo-stratified columnar ciliated epithelium. Some sites are mentioned here. Third type, look at this, beautiful. Either this one or this one. This is the one which is called as the respiratory epithelium. Somebody asked me, Dr. Zafar, are we supposed to know, memorize, remember all these photomicrographs? So the general answer is, we use these micrographs to confirm whatever is written, whatever text is written there. These slides are for your practice. I cannot explain without showing. So whatever I am explaining, you can confirm it by looking at the slide. You have to be careful. Okay. See, in this particular site, once again, you can see the polarity. You can see the presence of cilia. So this is pseudo-stratified columnar ciliated with goblet cells. Only this epithelium is found in the respiratory tract. It is called as respiratory epithelium. So the fourth one, and then I don't know, I didn't do anything. Look at this. This is once again pseudo-stratified, and look at this. This is the fourth. This is polarity. Once again, I am repeating, polarization means, polarity here means that the tendency of the cells to be present in one of the domain of the cell. Here it's basal 2/3. They occupy the base, they occupy the center, but none of the nuclei of any of the cell is present in the upper part of the cytoplasm of these cells. Okay, so this is pseudo-stratified columnar with stereocilia. Now, how to differentiate, how to identify cilia as cilia, how to differentiate stereocilia as stereocilia, or how to identify microvilli as microvilli. This is practical aspect. It needs practice, either in the lab or again and again, again and again and again and again. On Moodle, there is a presentation. It's my presentation. It's under the label Apical Membrane Specializations. It's a lecture. It's a presentation which I did not deliver. It is for your reference. If you want to know more about microvilli, cilia, and stereocilia, you can refer to that PowerPoint presentation. Is it clear? Okay. Now, this is polarity, and this is stereocilia as one of the surface specialization. Remember, if we have to differentiate a pseudo-stratified epithelium from a true stratified epithelium where the cells are arranged in many layers, we use these 2 points. Point number 1, pseudo-stratified epithelium shows polarity. You will not see any nucleus of any cell in the upper part of the cell. Number 1. Second, cilia, microvilli, and stereocilia are not associated with true stratified epithelium. The moment you see microvilli, the moment you see cilia, the moment you see stereocilia with this confusing epithelium, be sure that it is pseudo-stratified epithelium. It cannot be stratified because stratified is not associated with these specializations. Okay, so by this we finish our. Somebody has to be with me all the time. My 2 fingers are not working. I don't know why now. Please, can somebody come quickly? Faisal, you just stay with me. Even when I use my Mac at home, I use mouse. I'm not good at using this. Save my time. So I repeat once again, there are 2 key points, 2 important points to differentiate next presentation. Okay, quickly, now we are starting the second type of epithelium, which is stratified. I will read, and I hope most of the points will be clear. For any explanation, I am at your disposal. Now, it consists of 2 or more than 2 layers. So you have to start from 2, can be 3, can be 4, can be any number. Okay. In case of more layers, layers are named as basal, intermediate, and surface layers. I will explain. The basal layer. Now, basal layer means that layer of cells which rest on the basement membrane, or those cells which are touching the basement membrane. Okay, is usually cuboidal or columnar cells. Most of the time they are cuboidal, but can be columnar. Intermediate layer usually is polyhedral, polygonal, or maybe irregular shape. Now, the surface layer determines the name of the epithelium. If you remember, when I was giving you the criteria, I told you that the cell shape on the top is considered for naming the epithelium. So it can be squamous, it can be cuboidal, it can be columnar. We will see. Now, the basic difference between simple type of epithelium and stratified epithelium is that stratified ones in general are for protection. Simple ones are for absorption, for secretion, for diffusion, because they are single layered. You have to apply your brain. Okay. Now, it's written over here, it's poorly suited for absorption and secretion. The classification of stratified epithelium is based on the shape of the cells on the surface. Now we will start. Once again, they are of 5 types. We'll start from the most flat cells, stratified squamous. Then squamous covered by a layer of keratin. It becomes stratified squamous keratin. In case of stratified epithelium, the surface specialization is keratin, no cilia, no villi, no stereocilia. See, this is how we differentiate. Then the 4th is columnar, and the 5th is once again special type, like the pseudo-stratified in case of simple. Take them one by one. Okay, quickly look at this, a diagram, and compare it with the actual photomicrograph. Start with the basement membrane. This layer is called as the basal layer. It has to be one layer. Only one layer of cells can touch the basement membrane. In the drawing here, they are columnar, no problem. Then the second layer becomes polyhedral, polygonal, polyhedral, polygonal. As they go up, you can see this effect that by the time they are here, they become flat, they become flatter, they become flattest. Here they are the flattest cells. So if you are following me, few sentences, when they are here at the base, they are close to the source of nutrition. When they are close to the source of nutrition, they are active. When they are active, they enjoy their life. They enjoy their life means they all the time are doing khatakat, dividing. By what? Mitosis. Okay. They divide. The newly formed cells, they try to come into the second layer. When they come into the second layer, they push the already present cells in the second layer. The second layer becomes the third layer. Then once again division takes place. Once again there is a push from below. So the third becomes fourth, the fourth becomes fifth. So try to understand this movement of the cells. By the division, continuous division of the cells in the base or at the base, the upper layers are being pushed up, 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 up. And a time comes when they come onto the surface and they are thrown out of the epithelium. The moment a cell divides and gives rise to 2, the same number of cells are pushed out of the epithelium so that the total thickness remains the same. Otherwise, this epithelium will become the epithelium or epidermis of elephant. It becomes a disease. So this cycle of division from base to the surface is controlled. Try to understand. One of the students asked about this, Dr. Zafar, what do you mean by this maturation? Try to understand. They are here. When they are born, they come here. They mature. They become older. They mature. They become older. Older. Oldest. And then they become no more. They die and they are thrown out of the epithelium. So from below upwards is the process of maturation. So all these cells in these layers, they are actually coming from here. They're not new cells. They are the product of the cells here. Details later on. Consist of variable number of layers. The basal layer consists of a single layer of cuboidal cells. They are in a state of continuous division by mitosis. Okay. Intermediate layer consists of few layers of polyhedral, polygonal, or hexagonal shaped cells. Surface layer consists of 2 to 5 layers of flattened squamous cells. The change in the shape and size of the cells exhibit maturation from basal to surface layers. This is what I explained, and this is what was asked. I hope I answered. Who asked this question? So did you get the answer, my dear? Okay. Now, there are no blood vessels in the epithelium. So epithelium is an avascular tissue. What I normally say, these cells are close to the source of nutrition. That is why they can divide. They are always in the they are always in the state of division. And as they keep moving away from the source of nutrition, they are deprived of oxygen, deprived of nutrition. By the time they are here, they are actually dying and out of the epithelium. No more there. Because of this, as they are away, as they're moving away from the source of nutrition. Now it's easy to see, oh my God, this is the surface. Somewhere here is the basement membrane. This is connective tissue. See, that is why the nuclei here are dark. They are in the division. As they are moving away, look at these nuclei. These nuclei now look like the nuclei of the squamous cells. Try to understand this effect of maturation from base to the surface. A very, 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 very thick epithelium, basically, primarily, mainly for protection. How can you expect absorption from this epithelium? So now, wherever you need protection, you need to protect underlying tissue, the tissue number 2, connective tissue. Allah subhanahu wa ta'ala has provided us with a protective epithelium, that is a stratified type of epithelium to start with. Is that clear? Now, this is the stratified squamous. Now, it is of two types. You can classify it as questions at the end. Okay, my dear? Wait. Stratified squamous can be a dry type of epithelium and it can be a moist type of epithelium. This is my explanation. Okay. Now, skin is exposed to sunlight, to heat, to cold, to thermal, to mechanical, to radiational, to frictional. Oh my God, so many type of insults, bacteria, parasites, this, that, pressure. So skin is a dry epithelium. In order to protect the underlying layers, skin is covered by, I will show you, a layer of protein which is called as what? Keratin. So wherever there is a dry surface, stratified squamous epithelium somehow is covered by a layer of protein. It is called as what? Keratin. Keratin has a property, structural property to withstand, to tolerate dryness. I repeat, simple English word, keratin can tolerate dryness, no problem. Like an umbrella protecting the underlying cells from the effect of temperatures, heat, abrasions, frictions. Is that clear? But where there is no exposure to heat or sunlight, you see in the oral cavity, Allah subhanahu wa ta'ala has provided us with saliva. Yes or no? Saliva means all the time our oral cavity remains moist. But at the same time, we use our oral cavity with the help of tongue, with the help of teeth, with the help of cheeks, with the help of gum, with the help of palate for chewing. So is there friction or not? Yes or no? So what type of epithelium is required in the oral cavity? Simple or stratified? But the moist one or the dry one? So here we have stratified squamous epithelium without keratin. So simple. So stratified squamous epithelium can be with keratin, without keratin. Now I will read. Blood vessels are present in the connective tissue. This concept we already know. The basal cells are close to the source of nutrition. So see, everything is explained. As the cells move away from the source of nutrition, they lose their shape and size. This type of epithelium, the first one, cannot withstand. Withstand means tolerate. Desiccation means dryness. So it is always kept moist. It is always kept moist. Remember, stratified squamous epithelium, there has to be some secretion. The loss of surface cells can be replaced by the division of the cells in the base. I think I have already explained. Now, functions, the same scheme, the same methods, the same style of writing, and then some of the locations. Easy way of remembering this type of epithelium is, see, all the openings which are leading into some of the cavity of the body are provided with moist type of epithelium. I'm using this term moist because you are beginners, so that you can understand stratified squamous non-keratinized epithelium, because it is always kept moist. Now, what are the openings which lead to different cavities? Oral cavity, nasal cavity, cornea, ear, the pore of the sweat glands, sweat pores, the urethra, the anal canal, the vagina. These are all openings which are leading towards some of the cavity of the body. You can see this list is, instead of memorizing the list, just remember one word, doctor. Any opening that leads into the body, the first opening, the guard is stratified squamous non-keratinized epithelium. Now, make it stratified squamous keratinized. Look at this. Oh my God. This extra layer. Now, this is a process. Keratin is synthesized by a process which is not our discussion right now. Maybe at some other stage. Keratin right now, for the time being, is something that covers the already present stratified squamous epithelium to protect it from dryness. Full stop. Keratin is an additional thick layer. You all know keratin is here in our palms, our soles, you know, the skin that we remove. And the females, they know, they all the time, they go to this pedicure and manicure type of thing to remove this thing. So you actually try to remove this keratin from the skin so that your skin becomes smooth. If you compare men's hand with women's hand, if you compare your hand with the labor who is contributing in the construction of a building, their hands are very rough. Even you can see callus in their hands, you can see callus in their feet. This is all keratin. So keratin is over the skin. It makes non-keratinized epithelium keratinized. One very important function is to protect from the effect of dryness. Is that clear? Now, after explaining this, it has features same as the stratified squamous epithelium with following additional features. It is designed to tolerate dryness, so it does not need lubrication. A layer of protein called keratin is produced by the basal cells that cover the surface of the squamous cells. Keratin layer protects the underlying layers from the effects of 1, 2, 3, 4, 5, whatever. It is mainly found in the skin. Now, this point is important. I was coming to this point. In some organs, instead, in some places, stratified squamous epithelium, stratified squamous non-keratinized epithelium can be changed to keratinized epithelium. It means, like, this is oral cavity. The epithelium is stratified squamous, non-keratinized. But if somebody is in the habit of eating dry food, dry stuff all the time, using chewing gum, or the effect of friction is more and more and more and more, and there is no enough lubrication. So in order to protect the cells, the same epithelium can produce keratin to protect itself from that increased effect of friction. So non-keratinized can be converted into keratinized. Is that clear? All these will be repeated 100 times when we will discuss different organs. But this is, you can make it bold, you can make it red or whatever color so that you remember it. Now, this is just comparison. Remaining epithelium are very simple. Only two layers, basal and surface layer, stratified cuboidal. I will read, you will understand. It is a thin type of stratified epithelium, usually consists of 2 to 3 layers. Basal is cuboidal, surface is also cuboidal. Its name is stratified cuboidal. Its function is to provide a thick lining for ducts, mainly for the exocrine gland, so that they can bring secretion from one part to another part. Okay. It is found in larger ducts of the pancreas, salivary gland, and thyroid glands. Please help me. As I told you, there are some points which cannot be explained at this moment. So you have to accept them as it is, and then wait for that moment where you can apply this knowledge. Is that clear? So if there is an explanation required, I promise I will explain. I will not go forward. I will not move forward until and unless I explain it. It doesn't need any explanation. It's just the description that it's a thin type of stratified epithelium. 2 layers are there. The second is stratified column. The fourth one is stratified columnar. 2 layers, basal layer, and the surface layer is columnar. Can be 3, can be 4, can be 5 layers. You can see diagrammatically, and you can see here. Theoretically, if you are recording and this lecture is being recorded, theoretically, stratified cuboidal and stratified columnar type of epithelium are exactly the same. The sides, the functions, the locations are the same, except for when we see them under the microscope, we see the shape of the cell as columnar on the surface, and in case of the other, we see the cuboidal cell. Their sides are almost the same, they overlap, their functions are the same. So as such, there is no difference in the description. Now, this is the beauty. Transitional epithelium. Transitional epithelium. Once again, a type of stratified epithelium. So if you are following my lectures, there were 5 in the simple. The last one was special. Am I right? Now there are 5 types of stratified. The last one is special. What is special about it? Yes, we need to know. I will answer the most difficult question so that you understand. You first see the diagram. Then I will explain. It is a stratified epithelium. Point number 1 clear. It has a basal layer. Yes, it has a basal layer. It has a intermediate layer. Yes, there is an intermediate layer. Now this layer of the cells is called as the surface layer. Now let us see the shape of the cells in different layers. Here in this diagram, the basal is columnar, no problem, it can be. Intermediate are polygonal, no problem, possible. Now the difference is here, the surface layer. Are they squamous? They are not. Are they cuboidal? They look like, maybe. Are they columnar? Now, I'm going to tell you some features and then I'll explain here, then I will read. Now, the first layer is a basal layer, columnar cells tightly bound to each other. And of course, as usual, they have the ability to do khatakat. They can divide. Now, this layer of cells, which is maybe 3, 4, 5 layers, intermediate layers. Okay. Now, this is important. These cells in this layer are not tightly bound to each other, just touching each other. It's an important point. They're not tightly bound. They're just touching each other. Okay. So that, suppose if I hold this epithelium from sides like this, and I stretch it like this, like we stretch the rubber band or any rubber, like stretch it. When you will do like this, because these cells in this layer are loosely bound, they will move on sides. When they will move on sides, the fourth layer will become the third and the third become the second. Because they can move, the number of layers will increase or decrease. Thank you very much. You are good. I appreciate. You understand my point? Now, when you release, they will come back to their position. From 2, they become 3, maybe 4, maybe 5 layers. So now the number of layers will increase or decrease. Thank you very much. Now this layer, the surface layer, they are tightly bound to each other. Like, try to understand this. See, it comes from inside. Tightly, no gap at all. Not a single molecule can pass through the gap. Normally, the cells are tightly bound, but not very tightly bound. Here they are tightly bound. Okay. Now, when you stretch, what will be the effect on the shape of this cell? They become flat or they become tall. Now you release them, they become. So they keep on changing their shape. Which layer of cells keep on changing their shape? The top cells. Now, sometimes they look like cuboidal, sometimes they look like squamous. Now it's a confusion for us. I don't know what to name and how to classify this epithelium. When it is stretched, it is stratified, yes, but squamous. Why? Because the cells become, and when I release, they become cuboidal. It becomes stratified cuboidal. It keeps on changing its shape. Now, what features I explained? The basal layer is columnar, okay. Intermediate layer of cells, they are loosely bound to each other. Their number of layers can increase or decrease because of the effect of stretching and relaxing. The surface layers, they can be squamous in appearance, they can be round or cuboidal. So because of this confusion, I have given it this transitional. The name transitional means, you understand? In between. In between what? Sometimes stratified squamous and sometimes stratified cuboidal. Now I'll give you example. Do you know what is urinary bladder? It stores bile. Yes or no? So what does it store? Urine. So bladder is always full or always empty or like what? Now when bladder is full, and suppose this epithelium is there, what type of epithelium will be there when bladder is full, stretched? Stratified? Squamous. And when bladder is empty, cuboidal. Okay. Now this epithelium is exclusively, when we say exclusively, it means it is only and only and only and only and only and only found in the urinary tract. Only in the urinary tract. And urinary tract has only few organs, either in the renal pelvis, the upper part of the ureter, the urinary bladder, and the urethra. Full stop. Wherever there is urine, we have this type of epithelium. Because now coming to the last point, number 1, these cells are tightly bound. Number 1. Plus, look at their shape. They are like this, like this, like this. This appearance from top is called as an scalloping appearance, as if so many individuals are holding their umbrellas. And if you see from top, umbrella, 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 or in other words, doom-shaped or dome-shaped. They cannot hold the urine. They allow the urine to slip all the time. Plus, these cells are specially designed. They do not allow even a single drop of urine to be absorbed, because urine is very toxic. It is very acidic, right? So this epithelium is designed to tolerate the acidic effects of urine. It can hold urine, it can face urine for two important factors. Two important factors. Number one, because of the very tight junctions, they do not allow. And plus, I am going to explain here, and then please, later on you can ask here, this part of the plasma membrane is unduly thick. I repeat, the plasma membrane means the phospholipid bilayer. The molecules are bigger. The membrane itself is thick, plus all the organelles, if not all, most of the organelles of the cytoplasm are here. They are gathered, they are collected, they are sequestered here under the membrane to support it. So the upper part of the cytoplasm is very thin. All these effects collectively help the cells not allow the urine to be absorbed. What do we want in the urinary tract? We do not want the urine to be absorbed. Okay. What type of epithelium is there? Transitional. What are the characteristic features of the transitional epithelium? They do not allow, or it does not allow the absorption. How does it work? Two important factors. Number one, there are only two possibilities for a drop of urine, either to pass between the cells, not possible because of the junction, or to pass through the membrane, through the cytoplasm of the cell, as in case of the squamous cells, not possible because of the specially designed cells. You understand my point? I will read, you will understand. It's a special type of stratified epithelium. It is exclusively lining the urinary tract. It has some features in between stratified cuboidal and stratified squamous. Usually consists of 4, 5, 6, 7 layers. Sometimes when we quote these numbers, they are bookish. There can be variation from reference to reference. So don't make these issues. Basal cells are cuboidal to columnar. You can see in the diagram. Intermediate cells are polyhedral. Now this, you will see it's a text, but it's from a very good reference. I have copied it from the reference so that you have that information. See, first look at the diagram. Can you see this dark staining upper part of these cells? Yes or no? This is the effect I tried to explain, plus the cells are tightly bound. If you want to confirm the same effect under the light microscope, look at this. This is one cell, and see this part of the cytoplasm of the cell is staining darker than the rest of the cytoplasm because of the movement of the organelles to support the membrane. Surface cells are pale stained and are large umbrella or dome shaped. Each cell has a large round nucleus with a prominent nucleolus. I hope you understand what does it indicate, okay? With a prominent nucleolus, and some cells have two nuclei. The luminal plasma membrane is thick and the cytoplasm under the luminal plasma membrane is crowded with organelles. Both these features are responsible for the thickening of the luminal plasma membrane and the dense staining of the luminal plasma membrane. The surface cells are tightly joined to each other by means of intercellular junctions. All these features help to prevent from the toxic effects of urine. When you will read again and again, so that is why I say it's a special gift of Allah subhanahu wa ta'ala. The cells are otherwise very fragile structures. Now, this is just the explanation. In the relaxed, non-distended state, there are more layers. In the stretched, distended state, layers reduced to 2 to 3. It is because, and I do ask this question, it is because of the intermediate layer, the cells of which are loosely attached. I think I'm answering all otherwise difficult questions. Finish. Faisal, come quickly. If I can open third. I think I can open. No need. Thank you very much. I'm learning. Nobody, you have to come. Come again, Faisal. Do this exercise. Now, today my speed is good. That's the point. Okay. Now, glands. Basic concept. Okay. If you can understand this, my problem will be solved. No. The topic is glands or glandular epithelium. I'll start with this without showing you any slide. Glands. A gland is a secretory cell or group of cells. One individual cell can be, in its entity, can be an individual independent gland, or sometimes you need a group of cells to be considered as a gland. Origin. You will not understand. See, that is why I use diagrams. That is why I used photomicrographs to explain. I'm just reading, then I will explain. All glands originate from the epithelium, hence called glandular epithelium. Believe you me, if I simply read and do not explain, you will not understand anything, but now you will understand everything. Look at this diagram. You can see an epithelium here. This is surface. You have to be with me for this. This is surface. This is epithelium consisting of cells, and there is a line which is called as basement membrane. It can be simple epithelium, it can be stratified epithelium. I repeat, now go back to simple epithelium. You have to make your own glands. Okay, now take any epithelium, any epithelium. Now what happens? Sometimes these cells of the epithelium, they receive, sometimes they hear something and they receive a stimulus. Some factors come from somewhere, and information comes maybe from the master, the brain, through the blood. This is the connective tissue. Can you have blood vessels here in the connective tissue? Yes or no? Capillaries, blood vessels. Now, maybe some hormone is controlling them, or the brain is sending signals through the blood. Some signals come, these cells which were making an epithelium, and they knock like this. Cut. Come on, wake up. These cells of the epithelium, which are making a simple epithelium, they wake up like this, and they start doing cut. You know what is my cut now? Now, when they are stimulated, so for a stimulus, there has to be some signal, either a neuronal signal or a hormonal signal, or maybe somebody shouts, "Oh, you please start dividing." So they start dividing. There are two possibilities. Just few minutes before, I told you when I was explaining the stratified epithelium that the basal cells, when they divide, the newly formed cells are pushed into which layer? Second layer, and then second becomes. So what is the direction? Which direction they are growing? Up, and you can see it. But this time what happens? The newly formed cells, the two daughter cells, when they are born, they push the basement membrane down. Try to understand this effect here. They push what? Basement membrane down. And now this process is continuous. So you have to draw your own drawings. Drawing number 1, draw an epithelium, simple epithelium with intact basement membrane. Drawing number 2, two of the cells divide and they push down the basement membrane, so you can see 2-3 cells. Drawing number 3, the cells are dividing and these few layers become maybe 4-5 layers. Drawing number 4, maybe 8 layers. Drawing number 5, 6, 7, 8, and then finally you see this. Possible or not? This figure is maybe after so many divisions. Where is the epithelium? This is the epithelium. The basement membrane, this is the basement membrane. These cells are what? They are the product of these dividing cells. What are they doing? Doing khatakat. And now invaginating, growing deep into the connective tissue. You cannot see them from the surface. Can you see them from the surface? Can you see sweat gland from surface? But it opens where? On the surface. You are following me? So it keeps growing and keep growing and keep growing. So it is here. Now come here. I have given 2 colors. The same epithelium is here. The same epithelium is here. The pink color is here. The green color is here. The basement membrane is there. So here, if you see here, you do one thing. You please erase the cells in the center. Erase, because it's your own drawing. The moment you will erase, a space will be created here in the center, like this space. Possible or not? In real life, what happens? When some of the cells are dividing, the cells in the center, they die because their life is this much. They die, they degenerate, which is a normal process. How many fingers can be seen here? When the hand was developing, they were like this, fused together. Fused like a web of a duck. Imagine if these cells between the fingers, they do not die, they do not degenerate. We all will be having web. So how do we get these fingers? The cells between the fingers, they have to die. This is called as programmed death. These cells were ordered, programmed when they were born at the time of fertilization, that by the time you reach there, when you will be lying between the fingers, at this moment you will receive a signal and then you have to say goodbye. We have beautiful fingers. You understand? So These cells, which are here in the center, they die, we get this structure. Thank you very much. Discussion, explanation is over. Now, this structure, which develops, which takes its origin from the surface epithelium. Am I right or no? Its origin is from where? From the epithelium. It has two parts. The deeper part, the green color part we have given. These cells, they acquire the function of secretion. Whatever they secrete into this space, and this structure, which later on becomes a tube-like structure, it is called as the duct. It brings the secretion to the surface. This structure is called as the gland. So how are glands originated? From which tissue? How many parts do they have? Two, a secretory part and excretory part. Secretory part consists of secretory cells. Excretory part, which is also called as the duct. It is continuous with the surface epithelium. It brings the secretion. So initially, all glands of the body, with no exception, they take their origin like this. In some cases, what happens? The cells of the duct, by the same process, programmed death, they die. When they die, so there is no connection of the secretory cells with the surface. But remember, they are surrounded by the connective tissue beneath the basement membrane. So they secrete into what? Directly into the blood. These glands or these secretory cells, this gland become a ductless gland. Ductless. But was there a duct initially? They are also called as endocrine glands. They are endocrine, exocrine, with a duct, without a duct, with a duct, exocrine. How many parts? Secretory part, excretory part. Exocrine, endocrine glands, no duct. It doesn't mean that there was no duct. There was a duct. It dies, leaving behind only these secretory cells, which are surrounded by the blood. Secretion goes directly into. So my point is, all glands, they originate from epithelium. Now, classification: exocrine glands with duct, endocrine gland without duct. So it's so easy. Glands can be one cell, gland can be multicellular. The only example in the body of unicellular is a goblet cell, which is a modified columnar cell, separate topic. All other glands in the body are multicellular. Now, their secretion is of chemical nature. It can be mucus, it can be serous. So I'll explain only one point about this, no more details. The important point is that look at, if I have that diagram, let me check. Otherwise, okay. Just one point. Look at this. Serous. Look at this cell. Okay. Can you see these cells? Yes or no? Now, what is this color here? Pink or purple? And what is this color? No, this one. This is not pink, it means purple. After that, I will stop. I will take questions. Sometimes in some cells, and it's the answer to questions. So many of you are asking this question. Normally, nucleus stains purple or blue because it is acidic. It is stained by basic dye. It takes the color of basic dye. Good. Cytoplasm in general takes the pink color because it is acidophilic because of the proteins in the membrane. So it is stained by acidic dye. Now, sometimes there are cells especially designed for protein secretion. So the type of organelle which is abundant in their cytoplasm is rough endoplasmic reticulum. Which is reticulum? Rough. And rough is rough because it is studded with ribosomes. And ribosomes contain acid, ribonucleic acid, which is RNA. So if there is a cell which has more rough endoplasmic reticulum in its cytoplasm, that area of the cytoplasm will be stained by the same stain which stains the nucleus. So within the cytoplasm, you will see an area of basophilia. Rest of the cytoplasm will remain pink. But where there is endoplasmic reticulum, it will give you blue color. So there will be two colors in the cytoplasm. Two colors. One for the endoplasmic reticulum, basophilic, blue color, and the rest of the membrane is eosinophilic. So this is very important. These glands we will discuss in GIT and other places, but this concept of staining, I will ask, if you see in the cytoplasm pink color, what is the reason? if you see blue color in the cytoplasm, what is the reason? Is that clear? So I stop here. My objective was to give you the basic concept of glands. All type of glands, anywhere in the body, they have their origin from the epithelium, whether it is simple epithelium, whether it is stratified epithelium. Is it clear? Very simple. They can be exocrine, they can be endocrine, they can be unicellular, they can be multicellular. And according to their different shapes, see, there are different criteria in different on different occasions. I will be explaining these. So more important is the staining than the classification. Now I'll take your questions one by one. Yes, please. Goblet cell. I was expecting that Dr. Oni will explain. No problem. No problem. A goblet cell, imagine simple columnar epithelium, basement membrane and columnar cell. So in the columnar epithelium, simple columnar epithelium, some of the columnar cells modify themselves and they modify their structure and they become mucus, which is the type of secretion, mucus secretory cell. So do you drink alcohol? Why? Have you seen a goblet cell, a glass, wine glass? Wine glass. Nowadays, wherever you go in different restaurants, they serve you juices in those beautiful wine glasses, goblets, which you can hold and you can, as if you are drinking something. You understand what I'm saying? So they acquire the shape of that goblet. Okay. So it's an assignment for you. Read it. Any difficulty, complete. Yes. Any other question? See, polarity is a property, can be anywhere, but we take the help, like it becomes helpful for us when we have to differentiate between two epithelium. On one side, there is the stratified where the nuclei can be seen in the top layer, like you will not see any space there. But when you see a pseudo-stratified epithelium, you will see, oh my God, there is a distinct absence of nuclei in the upper layer. So this property becomes helpful for us there. Thank you and shabash. Before I forget, it's their right. Yes, yes. Can you come to the mic? You are already. So Faisal is here for some announcement and he has only 3 seconds. 1, 2.
Not me.
Farah, come. Who is the president?
There's no president.
Like you are for different.
Yes, one
Come on,
group.
quickly. Introduce
Oh,
yourself.
no, the committee heads, you mean?
Yes.
No, we're just the BLCs.
You are the BLCs. Okay. Year 1 BLC.
Yes.
They are your representatives. Okay. So ideally, how do we work for any of your problem, for the classes problem, instead of complaining individually, you can come through them. Okay, no problem. You please quickly before the.
Hello, my fellow batchmates. We are the elected BLCs. And first and foremost, I am Farah Abu Rab, not Abu Rab. I'm sadly not a godfather. You can call me Farah Atif, that's fine. And we are here to help you in everything that we can possibly do. And you can come to us with anything. My fellow BLCs will be elaborating more on the things that we are currently doing. And we are Ghaid, Faisal, Farah, and Muhammad.
Who is Faisal? Okay. Who is Ghaid? Who is Farah? And you are? Muhammad Uwais.
Hello, I'm Mer, as Farah has introduced, part of the BLCs. As you know, there are 4 committees: Committee of Lab, Notes, Exams, and Coordination. We're working together to arrange meetings. So any of the batch committee heads that have been appointed, please come after this class ends. We're going to arrange a meeting. And if anyone needs help, anyone at all, my number and email, both are up. Please contact me. Thank you so much. On to Ghaid.
Good. Congratulations.
Okay, so attendance is going to be our first action as official BLCs. I know we're all stressing over med school, so we don't need to stress over attendance as well. So we will be sending an attendance form. It's a Google form that you guys will be able to fill out every day in case you have problems with marking your attendance. You will have to fill in your name, ID, the class, time, as well as a screenshot of the message you get when you're unable to mark your attendance. Do you guys know what I'm talking about? You get like a red X whenever you can't. Okay, So we're going to need you to provide a screenshot just so we can make sure that we're actually here. The deadline for now to submit the form every day is going to be 5 p.m. Please try to meet this deadline. We plan to send one email to doctors to keep everything organized and simple. Thank you.
Assalamu alaikum. I'm Faisal, and for now, I'm recording the lectures. So no need for any of y'all to record them. Inshallah, I'm going to upload them on a Google Drive and send it to everyone. That's it. Thank you.
I think I congratulate them all, but like my request to you regarding the lectures, because sometimes when we are in a flow, there is a possibility of slip of tongue, or maybe some wrong figures. I already mentioned it before. If you notice any such thing, please bring it to my knowledge. We will correct it. Important thing is concept, understanding. Now, what's your question? Thank you and Shabash. So I'm expecting a party from them. Okay. Yes. So first task for you, BLC. Somehow you need the solution for this mic in the Princess Aya Auditorium. Yes.
Doctor, you mentioned that epithelial cells get their nutrition from underlying connective tissue. But what about the epithelium that lines cartilage? They're both avascular. How do they get there?
What about?
What about epithelium lining cartilage, which is an avascular connective tissue?
Which connective tissue is avascular?
Yes.
Cartilage.
Yes, cartilage.
Thank you for asking this. Cartilage is an avascular tissue, but when you will discuss cartilage in detail, cartilage structure is that it has an outer part and inner part. There is a membrane covering the cartilage, which is called as perichondrium. Am I right? In the perichondrium, there are blood vessels. So by the process of diffusion, the nutrients and oxygens are supplied to the inner part of cartilage. Number one. Sometimes cartilage are very thick. By the process of diffusion, or the diffusion alone is not sufficient to fulfill the requirement. So there are canals passing through the center of the cartilage, which are called as cartilage canals. And it contains blood vessels. So inside blood vessels, outside blood vessels. Once again, the process remains the same, the diffusion. That is why, because of the avascularity and the limited diffusion, the cartilage never grow too thick. They are thin structures. You understand my point? So thanks for asking this. It's a very good question, and shabash. I think now you all know my shabash. Yes, please, quickly. Polarity, I think I've been explaining it again and again. So those who have problem regarding polarity, please come to me in your free time. Then I will make it very clear to those who are still not clear. Okay? So your question is valid. On hold, come, spend some time with me. I'll explain by drawing myself. Oh, just write me an email. I'm not that big shot. I'm a simple man. And never close my door. And you will never find me free. So the best way is just come and enter. Or if you want to be very formal, write me an email. I'll tell you my free time. Okay, I think in this process, I don't want to take extra time. Dr. Abdullah's time is here. I think he's already here. So thank you very much. For TBL, whatever we have covered so far, it's included. So don't ask about TBL. Attendance, I did not take. Faisal, just come quickly. Abdullah, sorry. Abdullah is a very good friend of mine, like my younger brother. And day by day, he's becoming more handsome. Go back. Go back. Now you will see attendance here. And then final group. And the whole class today is 3rd of Sunday. I think this is open there quickly. Then no preference.
Which one?
Close September. What is this? Yes.
Or this one?
Copy attendance data from.
Okay.
Here. No. Generate. Okay, please.
Have a nice day.
How will you? No problem. Right now, we have some documents. Alhamdulillah, I was able to finish. So what I'll do now?
Generate 10 seconds.
Generate 10 seconds again. Okay. I'm generating again. Okay. 10 seconds. No, no, no. 20 seconds. Dr. Abdullah is here. He will mind, please.
No, it's okay. It's okay. They need the 5 minutes break.
Guys, I have generated the second key for those who missed for some reason, please. Even if you miss, write me an email. Thank you very much.
It's not working.
It's here.
No, it's not working.
So then send me an email. No problem.
Today?
No, yeah, anytime. Write me an email.
Don't worry, don't worry. We're gonna send you the attendance form and we'll send the doctors. All right. Alaikum salam. Alhamdulillah. ممكن تكلمني بعد؟